Live streaming is on.
Okay. So can you show my presentation? Can you see my presentation now?
No.
Or my whatever?
Do you have one screen or two?
Okay, I have two screens, I think. Okay.
Yeah, with two screens, sometimes it gives some issues.
Okay, let me see. So, okay. Now.
I think we are seeing, 1 second. Yeah, now we see your screen, like the one with the program.
Okay.
No, when like if you have the two screens, when you turn on the camera, I think it disappears. The other day happened the same. So I think you just have to share
I
the
should turn off the camera?
Well, when you start sharing, it turns off, so don't turn it on again, otherwise it stops sharing the screen. I don't know, something.
Okay,
with
okay.
That.
We'll see. Okay. Okay. And now?
1 second. We see your screen, but not the presentation.
Okay.
No, we see the website. Maybe you have to put the presentation in the other screen.
Okay.
What we see now is your internet platform on your Mac and your desktop.
Okay. Let me see. Okay. Oh my God. And now? Sorry?
We see a website showing like the profile of Cecilia, but that's the only...
Sorry. Well, I don't know.
Because where do you have your PowerPoint presentation?
Sorry?
Where do you have your PowerPoint presentation? Or
Okay, I have here in my computer, but I don't know if I should upload somewhere. Sorry.
No, you just have to open your PowerPoint as if you were going to present in your computer.
Okay. I open it.
We don't see it. Maybe you have to move it to the second screen, because maybe you're only sharing one of the screens.
Okay, let me see how I do it.
Did you have a pre-recording version?
Okay. I think here, maybe here.
Yes, we can see it.
Yeah. Right.
No, yes.
Sorry for that. Okay. Now you see it.
Yes, now it's...
Okay. So sorry for that.
Now.
And thank you. Thank you for the help, and thank you for the invitation.
Now it's not working again.
Oh. Okay, wait.
Yeah, I think if you disconnect the second screen and you use only one screen, like only like your laptop, if you're using your laptop, it's gonna be easier because you then don't have the two screens issue.
Wait.
There is one thing that I don't understand. It's that you are currently typing on your Dryfta platform, and it will be easier if you open PowerPoint on your computer and you directly click present on PowerPoint. Because here we see that you are in the profile of Cecilia, your colleague Cecilia.
Yeah, no.
David, I think also Cecilia is sharing her screen. That's the point. There are two
Okay.
people sharing the screen now, and
So we see the screen
Cecilia
of Cecilia at the same
Calleias
time as you.
Yeah, so
It seems that you are both on the same account. So there is a problem. You are two persons using the same account, apparently.
No,
Okay.
Cecilia is sharing the screen instead
Cecilia
of
is sharing the
Claudia.
screen. Okay.
So Cecilia, can you stop sharing screen?
Can we unmute? Or, kick her out.
Yeah.
Just a question, Cecilia, are you online? There is a problem. We are seeing your screen, Cecilia, and you should stop to share your screen, Cecilia.
I think if I start sharing my screen, Cecilia will stop sharing screen. So I'm gonna try that. So
Hola, Ceci.
now I'm sharing my screen. So I will stop.
De compartir tu pantalla.
And is Cecilia located in your lab? Is it the same group or not?
Claudia, you can start sharing now because now there is no one sharing the screen.
This is Murphy slow. Yes, perfect.
Now it's okay. Finally.
Okay. Right. And then you can see my presentation now?
Yes.
Yes, we see the title slides, and I hope that they will proceed.
Okay.
Screen.
Okay,
Yes.
and now?
Now it's okay.
Okay, thank you. Thank you very much. I'm sorry for all this trouble. Okay, I'm going to start with the presentation. Thank you to the organizer to invite me to give this talk. I'm going to talk about how to manage microbial community to produce energy from a Latin America perspective. So I'm going to talk from a Latin America perspective because here in Latin America, we have some characteristics that are maybe different from other parts of the world. And one of the characteristics is that our industry, most of our industry are agro-industry. And that's the wastewater treatment plants in general are separated from the sewage treatment because the industries are spread in the countryside. The other characteristic, but this more in the scientific community, is that we have a very important anaerobic digestion community. And maybe this is because we have a strong collaboration years ago with Professor Lettinga group. And we have a lot of methanogenic reactors here in Latin America, full scale. We have many USB reactors, also for sewage treatment. That this could be a little bit different from other parts of the world. And inside this anaerobic digestion community, we have a special Latin America biohydrogen network that is very, very active. And within this network, we are starting also a biorefinery network. So what we want in this network, we want to apply the biorefinery concept to wastewater treatment plants. This is a scheme of the wastewater treatment plant from an industry. You see the three parts of the treatment. And if we apply an anaerobic process in the secondary treatment, we can obtain many products as energy like methane, hydrogen, electricity, but also other products like organic acid, bioplastic, and so on. So our group of researchers from Latin America started to think in trying to convert our anaerobic digestion process to other processes. But this is not so easy, and I want to share with you this very, very interesting work that some years ago, that is the transition between environmental biotechnology to industrial biotechnology. And in this transition, we have to think that we are not going to work with one culture and one poor strain, but we are going to work with a mixed culture. But we want to manage this mixed culture. So we have to learn how to manage this mixed culture. So how we do that? In general, what we do is we run bioreactors at the beginning lab scale, but we can use also pilot or full scale. We take samples, we monitor this reactor, we take samples, study the microbial communities, and then we try to infer the function and then determine the best condition to select the appropriate microorganisms for the process that we want. I'm going to talk about two processes, methane production process and hydrogen production process. The methane process, we are studying in full-scale reactors in general, because this is a consolidated process, and I tell you, here in Latin America, we have a lot of application of this process. But we studied some related problems to the startup or also to the installed process. But for hydrogen, we are studying lab scale reactors, also few pilot scale, because we have still many problems to scale up this process. So I'm going to show you some examples of our work in USB full-scale reactors, methane production reactors. And this is a work of Cecilia Callejas. There we study a full-scale USB reactor that was installed in a dairy industry and milk processing industry here in Uruguay. And we have the opportunity to monitor this reactor from the installation, from the startup, from the inoculation, and during 1 year. And the idea of this work was to study what's happened with the microbial community during the startup of the reactor. But as in real life, we have a lot of problems, and one big problem that we have is that we have a spill of sodium hydroxide in the industry, and the pH of the wastewater increased to 9 for some days. So here I show you a picture, a big picture of the microbial community, samples taken during 1 year, and the analysis was done by 16S RNA gene sequencing. And you can see here a big picture because it's a level of phyla. And you see at the beginning, we have a diverse community, but the diversity goes down when we start to operate the reactor. But here it was the accident at around 100 days of operation. And then the pH of the reactor increased to 9, and then we have the opportunity to monitor what's happening in the microbial community. And you see here that we have a high dominance of one group of microorganisms, that was one Firmicutes, that is started to support this high pH in the reactor. But after some time, the communities get back to more stable community. So that shows us how this process is a robust process that could deal with some problems that often occur in the industries. And I'm going to talk about another work we are doing that is more specific in one group of microorganisms we are studying within methanogenic reactor, that is the chloroflexi group. Why we study this group? Because they are always present in all methanogenic reactors that we study. And we know by fish that they are filaments. And in a very nice picture you see here, there's a Sekiguchi work in 1999, and they show a fish picture showing the position of different groups of the microorganisms in a granule, in a USB granule, cutting the granule, and then you see in red the archaea, the methanogenic archaea, and in green the bacteria, and the chloroflexi bacteria are here. And you can see in this other picture, not so nice, but is a picture of the granules in a very, very unusual event of bulking in anaerobic systems. And you can see here the granules that start to have like filaments going up from the granules. And what's happening with that? The biomass, the density of the biomass goes down, and then the granules flotates, and then the granules goes out from the reactor, and the biomass inside the reactor decay, and we have a lot of problems. I have to re-inoculate the reactor. It was a full-scale reactor operated for many years. Then we do fish with these granules, and we found a very high proportion of filaments from the chloroflexi group. Then if we look at the bibliography, who are these microorganisms? They are a deep branch in the phylogenetic tree within the bacteria, and the taxonomy of this group is changing all the time because we have very few isolates. We know very, very little about this group. Nowadays, we have 7 classes according to Midas database. But for example, in the Anaerobic family, that is one of the most known families within this group, we have 8 described genera, 2 candidates, and 120 not yet described genera, according to Midas database. So we know very little about this group. That's why we focus in this group. And we start with our reactors. We have monitoring 5 full-scale reactors during 1 year, more than 1 year. And we especially studied this group by FISH, by qPCR, and by 16S RNA gene sequencing. And what we saw by FISH, that in the 5 reactors, they are filaments. And by qPCR, we see that the abundance is very high, but especially is high. Here we see the qPCR, the copy numbers by nanogram of DNA in the 5 different reactors, and the box plot are the samples of during 1 year. And you see that in these 3 reactors, the copy numbers are higher than in these 2, and these 3 reactors were operated for long time, more than 5 years when we started to take the samples, but these 2 reactors were operated from the startup when we monitor it. And the other result that we have when we analyze the sequencing with the sequence, we found that most of the sequence cluster in this cluster 1, we call, that are not related to any known genera, described genera, within the anaerolinear class. So most of the sequence cluster in this class, and most of the sequence cluster in a group with no culture representative. So after that work, we start to ask other questions. So what's happening in other reactors, in other full-scale reactors around the world, and which is the taxonomic position of this group, the abundance, and if we have any correlation with the operation conditions in this group. For that, to answer this question, we do raw data. Then,
Live streaming is on.
sorry, do you hear me?
Yeah.
Okay, sorry. So we have a lot of problems with starting this work to obtain the raw data. Then the other problem is that in this publication they use different primer sets, and the primer sets has different coverage for the group we are focused. That is the chloroflexi and the anaerobic. You see here in this table the different coverage according to the database. And this is one of the big limitations of our work. And this is one of the big limitations to do metadata analysis with published data. So, but we do this work and we analyze which were the chloroflexi that we found in these 62 samples in these 62 reactors. And in the blue box, you see the solid waste reactors, and in the red box, you see the abundance in the wastewater treatment reactors. And you see that most of the general what we recover within the chloroflexi groups were not described genera. And especially in this, it shows that there are some groups are that very important in all the reactors, but are not yet described. So what we are doing now, what we are trying to do metagenomes and ensembling genomes from metagenomes. You can see the poster of Patricia Bobbio today in the morning about which are the marks that we have and which is the function that we are trying to unravel with this. And we are planning to do metatranscriptomic to see which is the function that is going on into the full-scale reactors. Now I'm going to talk about the hydrogen. So how we obtain hydrogen. With the same process that we obtain methane, this is the anaerobic change for production of hydrogen of methane. One intermediate product is the hydrogen. And then we have to stop the last step, to inhibit the last step to produce hydrogen. And as we work for many years in methanogenic reactors, we know in which conditions methanogen doesn't work. Then we apply these conditions to inhibit the methanogenesis and to obtain hydrogen. So these conditions are high organic loading rate, low hydrogen retention times, and low pH in general. So, but within the fermenters, we want to have the microorganisms that can produce higher amount of hydrogen, like Clostridia, some Protobacteria, and some Termotogales. And we want to avoid the hydrogen consumers, the methanogens. We know how to avoid the methanogens. But there's another problem with other microorganisms, like homoacetogens, that consume hydrogen to produce acetate and CO2, and we don't know exactly how to avoid these microorganisms. And also we have a lot of competitors, other fermenters that produce other things that are not hydrogen, like lactic acid bacteria. So we started to have also a lot of problems for this process. And I'm going to show you two works that we have done with cheese whey. We used to work a lot in cheese whey because the production of cheese and milk here in Uruguay is very, very important. And cheese whey has a very interesting properties to produce hydrogen because it has low pH and high concentration on CO2. And we know it's not good for methane. And then we started to analyze these two processes, so methane, hydrogen and methane. And in the first work, we put low pH in the reactor and high organic loading rate, and we still have methane and hydrogen at the same time. And then we decided to use other inoculum, and it works better, but it's still, this is one of the last work. We use a CSTR reactor, and we have hydrogen, but the process is very unstable because at the end it was like consumed or goes down. So we want to know why the process is so unstable, because we can inhibit the methanogens, but still the process is unstable. And then we perform in this network, in the Biohydrogen Latin American network, we perform a shared work in studying the microbial community from 20 reactors, 30 samples taken in reactor from Mexico, Chile, Brazil, and Uruguay. And we analyzed the microbial community to see which is the microbial community in samples taken during high hydrogen production and samples taken from the reactors when the production is low. And that, well, in this picture, you can see the samples here, and here the genera, and in red color is the higher abundance, in blue is the lower abundance. And you can see here something that is not very usual in microbial community, that we can have some genera that are very, very abundant. Red one means more than 50%, and this dark red means more than 75% abundance of one genera. And we have samples like this, they have 4 genera, and one is more than 50. And if we see which is this genera, is Clostridium, so good. We have a good microbial community to produce hydrogen. But if you see this other, the yellow one, we have Lactobacillus that is not good for production of hydrogen. But when we analyze what's happened in the bad and good samples, we divided the samples in 3 groups. The group 1, it's the samples were taken during high production of hydrogen. The group 2, it was medium production of hydrogen, and group 3 is the samples taken during low production of hydrogen. And we analyzed the abundance of specific groups. We found that, for example, Clostridium is very abundant in this group 1, that's with high hydrogen production. But we found also the Lactobacillus is very high abundance. You see here, we have samples like more than 50% of Lactobacillus that works very good for the production of hydrogen. Then we ask ourselves, so why? What's happening with this? Because sometimes also we have high lactic acid bacteria and very low production. Then to answer this question, we isolate microorganisms from the reactors, and we isolate Clostridium, Lactobacillus, and these other microorganisms, Megasphaera, that the particularity of this group of bacteria, Megasphaera, is that they can grow on organic acid. And in this reactor, we have a lot of organic acid. And these microorganisms can grow on lactic acid and produce more hydrogen. And the conclusion of this work that we started to co-culture the different microorganisms is that when we co-culture Clostridium with Lactobacillus, we have complete inhibition of hydrogen. But when we co-culture with Megasphaera, the hydrogen goes up. So the conclusion is that we have a kind of interaction between these 3 groups of fermenters that in some conditions can increase the production of hydrogen, but in some conditions, the lactobacillus can grow up and the hydrogen production was inhibited. So we also performed this review within the network and trying to understand which are the problems associated to the instability of hydrogen production. And it was very interesting because we think about how to detect each of the problems. So for the methane oceans, it's very easy because we measure methane and we know by 16S that we have archaea methanogens. But for other group of microorganisms like homocetogenic bacteria, it's not so easy because some homocetogenic bacteria can also produce hydrogen and in some conditions can consume the hydrogen. So we do also another work specifically to study homocetogens by qPCR. That is, we are going to publish very soon. So what are the limitations of our works? So we have the limitation in trying to characterize the microorganisms because, as I tell you, with the chloroflexis, sometimes we have this sequence, but we don't know which are these genera and what they are doing. So we have difficulty to infer the function, and also we have difficulties from if we know the microorganisms and we also know the function, but we don't know in which condition we can grow the microorganisms, like homocetogenesis, homocetogenic bacteria. We can detect, but we are not very sure in which condition they can compete or they not. So some things that I think it could be nice to do within the MIWI community is to have a specific database for anaerobic wastewater treatment systems like MIDAS. Maybe MIDAS is expanding in the anaerobic world. So it's very nice to have functional database. I found acetobase is a functional genes database for acetogenic bacteria, and this is very, very nice to have because we can track specific microorganisms with this database. So, but we also need metadata standardization to do meta-analysis. We need to have more standardized the metadata available, and we need more work in ensemble genomes and functional information. So thank you very much. This is my group, and we have 3 posters from my group. Patricia is presenting a poster in chloroflexi, Ines a poster in lactic acid, and Cecilia in methanogens. So thank you very much, and thank you to be here, and sorry for all the problems that we have.
Okay, thank you for the presentation and the insights. Do people online or here in the room have questions for Claudia?
Okay.
Is there anyone? So you can put in the chat, or you can, if you want, just open your microphone and ask a question if you're online. I have to look. There's no. Maybe from my side, could you elaborate on what you now think is the ecological or physiological role of chloroflexi within the community?
Yeah. There are some hypotheses that we are trying to also to see if there are, because of the function. There are some macrobial ensemble genomes, and there are some isolate. They are fermenter, they are hydrolytic bacteria, and all are filaments. So our hypothesis, and also from other researchers, that is that they are involved in maintaining the biomass inside the reactors, just organizing the granules, or also in the floccules, and maybe this is why we have a higher abundance in USB reactors compared with solid reactors. In general, we have higher abundance in USB in methanogenic reactor for wastewater, that most of these reactors are USB or EGCB, so they have granules. And we have higher abundance in this that compared when we compare with solid waste that are CST or other kind of reactors. They're also very present in activated sludge, and we are trying to do some work in trying to compare the filaments. There are also filaments. We are doing some work comparing the chloroflexi that we have in activated sludge and also in methanogenic, and in anamox reactors also we have. And we don't know if they have all the same function or not, but it's probably related to the degradation of biomass or something linking to hydrolysis of some compounds that are always in the reactor that has high amount of biomass.
Yeah, so in my view, they are more or less either scavenging on exudates from microorganisms or growing on dead biomass. And if you then compare, for instance, a sludge digester or USB granules, the solid retention time in USB granules is much longer than in, say, digesters, which means there's also much more time and space for bacteria to grow on decaying biomass. So wouldn't the chloroflexi not directly be involved in the granulation itself, but just being there more dominant simply because of the longer SRT instead of forming the backbone of the granule in one way or another.
Yeah, this is an interesting topic because, as I show you, when we compare the USCB that were more long time operated, many years, we have higher abundance than when we start up. So this could be, yes, that they are maintaining inside the biomass or inside or outside, I don't know, maybe outside or whatever. And because they are filaments and they can grow also in biomass decay. But the question is, what's happened when we have bulking? Because they produce also bulking in activated sludge. Yeah, it's also produced bulking. But in anaerobic bulking, we don't know why it's occurs, you know.
Okay. Thank you for this question, for answering this question. Was there any on floor or?
There is a question from Professor Tom Curtis. Do we know which compounds they hydrolyze?
Yeah, we have some idea because, as I told you, there's some isolates and there are some also microbial assembly genomes. Also, we have some microbial assembly genomes, and they can hydrolyze several compounds, like, I don't know, whatever. Well, it depends of I think it depends of the genera, but they can hydrolyze proteins, they can hydrolyze polysaccharides, and we also studied the hydrolysis of some compounds that are in the cells, and they can hydrolyze. So they are not very specific, I think. So because we find that the problem is that we know that they have several genes for hydrolysis, but we want to know if they are active or not, or if the same genes are active in the different reactors or not. That's we are trying to see. But the big question is what happened during the bulking problems, why they started to overgrow, and we have not an answer. But bulking problem is always a problem.
Okay, well, thank you. The rest, if there were more questions, we will save them for the round table. So please stay online, and we'll see you later back on screen.
Okay, thank you.
Here in the room, we see all the, on this big screen, only the initials of people. I think from our side of the line, you can put up your cameras and we see your faces, because the bandwidth here should be big enough. If on your side that's a problem, then keep your webcam off. But if possible, just put them on so we can see also some faces here besides the initials. Then I will give the rest of the chairing of this session to Sergio Tomà, who can go ahead.
Yeah, let's continue with the oral presentations. So the first one is going to be Simona Crognale from the IRSA Water Research Institute, that is going to present structural composition and metabolic activities of the microbiome involved in the production of N-caproate from food waste.
I'm a researcher from the Water Research Institute of the Italian National Research Council. And today I will talk about the structural composition and metabolic activities of the microbiome involved in the production of caproate from food waste. Overall, organic waste accounts for more than 34% of municipal waste, and around 60% is composed by food waste, considering that 88 million tons of food are wasted every year along the entire food chain. In order to avoid the waste and in application of a circular economy strategy, during last year, the biorefinery concept has grown up, referring to the sustainable processing of biomass into a spectrum of bio-based products and bioenergy. Among all of the bio-based products, I want to focus our attention on the medium-chain fatty acids, that are fatty acids containing 6 to 12 carbons. And their production from waste has several benefits due to the renewable properties, the high economic value, and the several industrial applications. Among all of the medium-chain fatty acids, caproic acid, for example, has a market size of 25,000 tons per year, with a market value of around $3,000 per ton, and several applications in pharmaceutical, food, and chemical industry. The production of caproic acid from food waste is possible by the activity of microorganisms through a chain elongation process that is mediated by microorganisms and basically consists in the use of acetyl coenzyme A molecule for the production primarily of short-chain fatty acids as well as the acetate. And then the acetate is used along to butyrate through the reverse beta oxidation process. And in a second step of the reverse beta oxidation, butyrate is along to caproate. The process starts with the use of electron donors. The most studied electron donors are ethanol and lactate. And this is the process for the production of the medium-chain fatty acids. In this work, we have decided to use a real waste, that is the extract of food waste, very rich in bioavailable sugars, in a acidogenic fermentation process with a 3-liter anaerobic bioreactor with a pH control at 6 value in a mesophilic temperature and an hydraulic rotation time of 4 days. We have applied different organic load rates in order to assess the influence of the caproic acid production. And it is worth noting that this is a single-stage process without the addition of external donors, as previously performed in several studies. At the beginning, we have decided to apply a continuous feeding strategy. So basically, we have fed the reactor every day from Monday to Thursday. And we have observed differences in terms of caproic acid production in the reactor with different applied organic load rates, and the highest concentration of caproic acid was produced in the reactor with the highest organic load rate. From a logical point of view, the 16S amplicon sequencing reveals differences between the different applied organic load rates. In particular, the occurrence of bacteria known for the capability to produce lactate and medium-chain fatty acids from lactate and sugar was observed in both conditions, with the caproate producer Pseudoramibacter more abundant in the reactor with a 15 organic load rate applied. We have also analyzed the 18S region in order to verify the occurrence of yeast potentially involved in ethanol production that can be used as electron donors. But the results highlight the occurrence of eukaryotes most likely associated to the feed due to their non-use in cheese production and their pathogenic activity against fruits and vegetables. We have further analyzed the biomass, showing the highest caproate production through the shotgun metagenomic sequencing. The results suggest a production of acetyl coenzyme A molecule that is the key intermediate in the reverse beta oxidation pathway, primarily from lactate. Also, the enzymes involved in lactate and medium-chain fatty acid production were hosted by most of the reconstructed beans. In particular, the bean 1, taxonomically classified as Pseudoramibacter lactolyticus, is confirmed as the potential major contributor to caproic production in this system. So, coming back to one of my previous slides, metagenomic data in line with process data support and strongly support the occurrence of a lactate-based chain elongation process in our system. In a second step, in order to further improve caproate production, we have decided to use the same experimental conditions with the best organic load rate of 15 grams COD liter per day, but changing the feeding strategy. In this case, we have fed the reactor in a discontinuous mode. So we have fed only on Monday and Thursday. And the applied feeding strategy affected the production of caproic acid, and around 2 times more caproate was produced with a discontinuous feeding. Also, in the case of 16S amplicon sequencing, revealed the occurrence of bacteria able to produce lactate and medium-chain fatty acids. But the presence of caprate productions, that is one of the most studied caprate producer, was observed in the reactor with the best performance. And this is one of the first evidence of this caprate producing bacteria in a system fed with real food waste, real waste in general. So in conclusion, the study highlights the feasibility of the use of real food waste for the production of medium-chain fatty acids in a single-stage fermentation process without the addition of external electron donors. The applied organic load arrays and the feeding strategy strongly influence the caprate production in terms of both rate and yield. Also, a caprate producing mixed microbial community was quickly selected, with caprate productions associated with the highest caprate production. Metagenomic evidences, in line with process data, fully support the establishment of a stable long-term lactate-based chain elongation process in our system. To thank all of my colleagues at the Water Research Institute for their collaboration in this work, and of course, I want to thank you for your kind attention and for being green.
Thank you for the presentation. Is there any question in the? I don't see any question in the chat, but maybe in the room. Yeah, there is a question, so you can walk to the microphone.
Hi, Simona. Thank you for the interesting presentation. Looking from a practical point of view, I really like the idea, because how circular can you be if you apply this in practice? I was thinking, what would, according to you, be the largest challenge to tackle going from a lab scale to, let's say, a larger field scale?
Yeah, okay, thank you for the question. It's very interesting because we are trying to approach to a higher level. And yes, the challenge should be at the beginning the manipulation of the food waste, because we have collect the food waste and then centrifuge, so squeeze, and then centrifuge in order to divide the two parts, the liquid part and the solid part. So this is one of the most challenging activity, I think, because we are working on something like few kilograms, but when we talk about a higher level, we are talking about a very much quantity of food to treat. And then another point, a key point, is the management of the reactor, of the treatment, because we have controlled the pH continuously, and so this is a cost if we think in a larger scale. And also, another crucial point that we are trying to assess is the extraction of the caprate produced. So in order to use it in the industries, but also in order to avoid the inhibition of the bacterial activity due to the large amount of these compounds that can be also toxic at high levels. So I think that there are crucial points that we have to assess before to move to a larger scale. But it's very interesting, and our results are so very interesting also in from an economic point of view, because we can obtain a high value products from a waste, and it's very important in our world, I think.
Thank you so much.
Thank you for your question.
And we have another question in the Q&A from Sohail Neshat. Have you considered using metatranscriptomics to see if the genes of interest are being actually expressed from those genome bins?
Yes, thank you for the question. It's interesting also, and I'm analyzing the metatranscriptome data, and fortunately I have a little idea about the results, but I have not complete the analysis. So this is one of my big interest now, because we have data from the metagenomic, and so we have more or less which can be the possible pathway within our system, but I would like to assess the exact pathway that occurs, not only for the reverse beta oxidation and caprate production, but also about the fermentation, so from the sugars and for also hydrogen production. So it's working. We are working on it. Thank you for the question.
Thank you. There are some more questions that now I will send to you, so then you can answer in the chat. So
Thank you.
now our next presenter is Merve Atasoy from KTH Royal Institute of Technology, and which will present manipulation of mixed microbial culture by augmentation for desired volatile fatty acid composition. I think she's going to present live, so you can start sharing your screen. Are you there?
Yeah, I'm here. I'm sharing. It says it's failed, but okay. Sorry, maybe I need to give permission to the system for this.
What do you have as an issue with your sharing? So what happens when you click the button share screen? Rejoin. Yeah, virtual events always take our passion. It's just normal, and it takes 3 times more time than we need, and we have buffer time with the round table, so it should be okay. Let's move to the next.
Yeah, while we solve that, we are gonna move to the next presenter. That is Christelle Bouneme-Sawaya from Lebanese American University, that will present effect of membrane biofilms and transmembrane velocity in an anaerobic membrane bioreactor on effluent quality and dissolved methane. And she's presenting online as well.
Hello, sorry, everyone. It's just gone. I'm again sharing.
So,
okay.
Merve could share it. So okay, so let's skip to the initial planning. So yeah, you can present.
Okay, sorry, it just there was a problem. Yeah. Okay, now
we see your screen. Yeah.
Oh, okay. Do you see it? The full screen now? Can you see? Okay, perfect. Oh, thank you. Good afternoon, everyone. Today I will talk about how we can produce desired volatile fatty acid composition by manipulating the mixed microbial culture by using bioaugmentation method. I'm Merve. I'm working as a postdoctoral researcher at KTH Royal Institute of Technology. And my presentation includes background of the study, why we carried out this study, how we did it, and briefly our results. Actually, it was my PhD project, and today I will present only some part of it, so it will be kind of summary. So this is very well-known anaerobic digestion pathway, and with the recent studies, and especially growing attention to the carboxylate platform, now it goes beyond biogas production to volatile fatty acid production, because volatile fatty acid, in short VFA, can be used as a carbon source for the production of other bio-based products such as bioplastic, biogas, or longer chain fatty acids, or we can use them as a raw material in the downstream processes in different industries such as pharmaceutical, chemical, etc. And because of this, they have high market demand. And unfortunately, currently we are only demanding this market, supplying this market demand only 3% by bio-based techniques. Because in the system, there are many limitations, which includes limited production yield, unstable acid composition, and several problems after production of BFA in terms of separation and purification. And if we want to increase the yield, then we need to use pure substrate. It means high substrate cost, and of course, there are several process dilemmas in the system. But of course, as I mentioned previously, there are several studies is ongoing, and they mostly focus on to optimize operation environmental conditions and to using different techniques and methods to solve these problems. So earlier to this study, I also work on to optimization of environmental and operational conditions, such as different pH, using different reactor type, substrate type, different inoculum type. But besides of this, we also applied bioaugmentation to produce one type VFA composition. And bioaugmentation, it's a technique to enrich the existing microbial community by adding pure culture, co-culture, or another mixed culture, depending on our purpose. So basically, how we conduct this study, we use cheese production wastewater. Our pH was pH 10, alkali conditions. As an inoculum, we use anaerobic granular seed sludge. And we monitor soluble total COD, VFA composition, pH, and we had two different approach for bacterial community. One of them was microbial community analysis by next generation sequencing, 16S rRNA sequencing, and the second approach was quantification of the selected pure cultures in the mixed culture after bioaugmentation. So we conduct this study in the anaerobic sequencing batch reactors at 35 centigrade degree, and for phases, one cycle was 24 hours. And briefly, our bioaugmentation strategy. We selected 3 different pure cultures depending on their metabolic pathways, and we selected these also based on our previous results. And Clostridium bitricum for bitric acid production, Clostridium aceticum for acetic acid production, and Propionibacterium acidopropionge for propionic acid production. So we bioaugmented each pure culture separately to the mixed culture during 7 days. Daily doses, we added each day 10% of the active volume. It was around 140 milliliters. And then after bioaugmentation, we operated our reactors for 2 sludges to monitor either they attach to the mixed culture or they wash out. So when we look at the results, actually, this one, bitric acid production from the Clostridium bitricum reactor. And here, the highest bitric acid concentration was around 3,000 milligrams COD after bioaugmentation. And at the same day, at the control reactor, and by the way, I forgot to mention about control reactor, which was not bioaugmented, but operated under the same conditions. And there, the bitric acid concentration, it was around 260 milligrams COD per liter. And when we look at the average bitric acid concentration after bioaugmentation at the bioaugmented reactor, it was around 1,600 milligrams COD per liter. And at the control reactor, it was around 300 milligrams COD. So our results show that as maximum, we increase 11 times, and as average, it was 6 times bitric acid concentration increased by bioaugmentation. And when we look at the Clostridium aceticum, the results show that similar to the bitric acid set, acetic acid concentration at the highest point, it was 1,200 milligrams COD per liter, and at the same day at the control reactor, it was around 130 milligrams COD per liter. And when we look at the average value, acetic acid concentration was around 836 milligrams COD after bioaugmentation at the bioaugmented reactor. And on the other hand, at the control reactor, it was around 200 milligrams COD. So also these results briefly show that as maximum 10 times and as average 4 times, acetic acid concentration increased by bioaugmentation. And when we look at the Propionibacterium set for propionic acid production, it was actually a bit different than the previous results. Their propionic acid production, it was around 1,000 milligrams COD on the maximum day at the bioaugmented reactor, and at the bioaugmented reactor, it was around 4,000. And at the control reactor, it was around 1,000. And when we look at the average after bioaugmentation, at the bioaugmented reactor, it was around 2,500 milligrams COD per liter. And at the control reactor, it was around 600 milligrams COD. And by the way, since our substrate was dairy industry wastewater, cheese production wastewater, our dominant acid type at the control reactor, it was also propionic acid. So after bioaugmentation, propionic acid production as average and maximum increased around 4 times. When we look at the VFA composition, the bioaugmentation effect was much more different than our expectation. So propionic acid was dominant at the control reactor with 60% dominancy. And after bioaugmentation by Clostridium butyric acid was dominant. And also these results showed us by bioaugmentation, we change not only VFA composition, but also acid dominance, acid type, which acid type dominant in the VFA mixture. But on the other hand, when we look at the acetic acid set, there even the acetic acid production increased around 10 times, but still propionic acid was dominant in the system. And in a similar manner, by Propionibacterium as the propionidae, propionic acid increased, but it was not very significant increase actually. So these results showed us, even though bioaugmentation strategy increased the target acid type, but it also enhanced other intermediate and end products production in the system. And therefore, we also wanted to see bacterial community profile. But there, since the presentation time is limited, I just want to say that the bioaugmentation changed bacterial community profile significantly. And if you like to hear more about the effect of bioaugmentation on the bacterial community profile, you can check the papers here, which were recently published. And the result of the study actually showed us bioaugmentation indicates certain unknown centrifugal relations and corresponding metabolic pathways. So as a conclusion, our study showed that, yeah, bioaugmentation is a strong approach to manipulate acid type and bacterial community profile. However, when we are designing this kind of strategy, we have to consider about its effect on functions and interactions of the microbial community, and at the same time we have to identify the metabolic network to understand the big picture. Thank you so much. If you have any question, don't hesitate to email me.
Thank you. We have time maybe for a short question. And I saw in the Q&A a question by Dr. Adel Lazuka. Maybe you want to ask it yourself? You can unmute and ask.
Hello. Can you hear me?
Yeah, I can hear you.
Good. Thank you. My question was about, can you please give more information about control reactors you mentioned? Was it considering the effect of media addition, or
Yeah, sure. At the control reactor, actually, everything was same with the bioaugmented reactor, only we didn't bioaugment the mixed microbial culture. It is operated under the same conditions. It is fed by the same substrate. Just we didn't bioaugment to understand the effect of bioaugmentation.
Okay, so the media was included.
Yeah.
Good, thank you. And maybe if I can, one more question was, did you investigate the downstream processing cost for such VFA recovery?
Unfortunately, for recovery part, we didn't do any study afterwards. Okay. But we know that it's very challenging.
Yes, thank you.
You're welcome.
Thank you. And we can move to the next presentation that I introduced before. So, Christelle, if you can start sharing your screen so you can already start with the presentation.
Hi, yes, I will share my screen.
Yeah, we can see.
Okay, you can see it.
Yes.
I'll just go back and make sure. Okay. You can see my screen, right?
Yes, and we can see you.
All right, perfect. So hello everyone, and my name is Christelle, and I'll be sharing our findings from our latest study, where we devised operational strategies to promote the development of membrane biofilms with minimal effect of transmembrane pressures in anaerobic membrane bioreactors. Now, anaerobic membrane bioreactors are known to be an emerging technology for wastewater treatment, and that because of the advantages they confer. However, they have current limitations in terms of membrane fouling and dissolved effluent methane. And so why are we interested in biofilms specifically? And that because they develop on membranes that are key component of ANMBRs. Not only this, anaerobic biofilms have distinctive mechanisms when compared to aerobic biofilms, and that in terms of the microbial groups that develop on them, the membrane microbiomes, and the extracellular polymeric substances that constitute the biofilm. Not only this, microbial communities has shown to be different on the attached biomass than the suspended biomass. And these groups are related to syntrophs, methanogens, and other genera such as Geobacter. And in a previous study, high relative similarity between attached ceramic carriers and in-reactor ones were highly similar to the ones on membrane biofilms. Moving on, in our latest study, we observed that key membrane microbial groups that developed on the membrane biofilm had higher relative activities than in the reactor. Not only this, they were linked to enhanced antibiotic removal rates. So this motivated us in our next study to develop and operate biofilms, doing so in a sustainable way. Also, in a previous, in a recently published review paper, those microbial key groups, such as methanogens, syntrophs, and exoelectrogens, were linked to enhancing emerging contaminants removal, such as organic micropollutants and antibiotic resistance genes. So our aim was to enrich anaerobic membrane biofilm microbial communities, doing so without increasing transmembrane pressure. And among our objectives were to determine a combination of different membrane biofilm development rates and transmembrane velocity, and that to see the effect on effluent methane, water quality, and antibiotic removal. So in our first phase, we set up the reactors with two membranes. Membrane A was allowed to develop the biofilm at a low pressure, whereas membrane B developed normally. For microbial communities, samples were taken frequently from the biomass, the membranes, and the permeates. As for the EPS characterization, whenever we harvested the membranes, the biofilm layer was differentiated into two layers, a loose layer that is easily scraped off the surface of the membrane, and another tight layer that is tight to the membrane. After the development stage, we operated membrane A with a new membrane for comparison purposes. However, during this stage, membrane A had higher transmembrane pressure than that of a new membrane, and so the pre-development of the membrane biofilm at low pressure wasn't successful. However, looking at the microbial community relative abundances, we see that we had main genera of Methanocore spiculum among all samples, as well as genera related to Centrophaceae. Also, what we noticed is that the microbial community on the tight layer of a new membrane was highly similar to that of a loose layer of a normally developed membrane, which told us that initial colonizers from the tight layer might end up on the loose layer. However, this wasn't evaluated in terms of performance in here. So moving on, what we tried to do in the second phase, doing the opposite, where we set up 3 membranes and we allowed the third membrane to develop at high pressure. All other operations were the same as phase 1. After that, during the operation stage, we let the 3 membranes operate at the same flux rate. And here we saw that the membrane E that was allowed to develop the biofilm at a high pressure operated at a similar transmembrane pressure range compared to the other ones. And so the pre-development of the membrane biofilm at high pressure was successful. We illustrate here the transmembrane pressure and the total membrane resistance profiles. And we noticed that during the operation stage, membrane that developed at high pressure was able to maintain the low transmembrane pressure during the operation stage. Not only this, what we observed is that during this same period, the membrane that we see here to the right was able to maintain the high total protein to total carbs ratio for both the tight layer and the loose layer. Also, whenever we check the performance of this membrane, we notice here that it's related to the lowest affluent COD, even though this had higher affluent methane. And whenever we looked into the trimethoprim removal, which is a common antibiotic, even though during the development stage we had similar affluent concentrations for all membranes, membrane that was developed at high pressure during the operation stage had the lowest concentration. And so this tells us that the membrane biofilm developed initially at high flux rate performed better in terms of trimethoprim and COD removals. Now, looking at the microbial community's relative abundances, here we're looking at 5% and higher. We distinctively see that the tight layer of the membrane that developed at high pressure had unique groups when compared to the biomass and the other membranes. What we were also interested in to look into this membrane compared to the loose layer and even at lower abundances of less than 5%. And also here we saw some unique groups that dominate the tightly bound layer. And this suggested that the high pressure could have some effect and shifted some key microbial groups. As a summary, membrane biofilms and anaerobic membrane bioreactors can increase degradability. The biofilms that pre-developed under high pressure can be recovered at low pressure while maintaining and without hindering the performance of the ANMBRs. However, methane dissolved should be and remain to be recovered downstream. Also, for the higher protein to carbohydrate ratios generated by the biofilm microbial communities may be linked to achieving enhanced removal under specific operating conditions. Also, this has been previously seen to be a negative effect. Finally, the microbial communities dominant in the biofilms likely influence EPS properties and aid in biotransformation during moderate transmembrane flux. This concludes my presentation. I would like to thank everyone who worked on this project and specifically my advisor, Dr. Mustafa Harib. And this research was in support with the LAU, Lebanese American University Graduate Program Scholarship, and PRIMA, Partnership for Research and Innovation in the Mediterranean Area. And thank you, everyone, and for the organizers as well.
And thank you for the presentation. There is time for some questions. So if anyone has questions in the chat or in the QA or in the live session. And I have one. You said that when you analyze the EPS that you had like two layers, like one that was more tight and the other one more like, I guess, loose. And did you see differences between those, like in composition or?
Yes, exactly. What we observed is that even between the tight and the loose layer, there were unique groups and different relative abundances, just as if I can reshare my screen. Even then, there were differences among the microbial groups that dominated the different layers.
And also in composition of EPS, and like what type of EPS analysis did you do? Only like carbohydrates and proteins, or did you go more in depth?
Yes, we only had the EPS characterization in terms of total proteins and total carbs.
Okay, thank you. Yeah, David?
Short question. What can be the effect of pressure on microbial selection? So what do you think is the underlying mechanism that would select for one or the other organism?
Yes, it seems that the high pressure that we allowed the membrane to develop at had pushed some key microbial communities to be more abundant in terms of exoelectrogens, syntrophs, and they might play a role in terms of enhancing biodegradation.
Thank you.
Okay, thank you. So let's go to the next presenter, and Cristina Gagliano from Wetsus, and that we will launch the video about proteinaceous substrate enhanced microbial granulation during anaerobic digestion in saline conditions.
Hello, everybody. Thanks for the introduction. Today I present this work, which I did when I was a postdoc at Laboratory of Microbiology of Wageningen University within the project Water.
One second, we are having problems with the audio. Cristina, do you still want to launch the video, or are you still having network issues?
Today I present this work, which I did when I was a postdoc at Laboratory of Microbiology of Wageningen University within the project Water Nexus, which we did in collaboration of the Department of Environmental Technology, exploring ways to treat saline wastewater anaerobically. The technology we did apply is the Upflow Anaerobic Just Blanket Reactor, and it is currently applied also large scale to treat high-strength industrial wastewater because of its ability to retain high biomass concentrations in the form of granules. And so this property makes this technology very suitable to treat highly saline wastewater. But pay attention, because if we would use freshwater granules to treat saline wastewater, what would happen would be granules disintegration, because sodium ions would have a detrimental effect not only on the cells for osmotic stress, which will die, but also to the extracellular matrix of the granules, where monovalent cations like sodium can displace the divalent cations in the matrix. The extracellular matrix of granules is indeed made by extracellular polymeric substance secreted by the aggregated microbial community, which is rich in protein and polysaccharides that can establish between each other different kind of chemical weak interactions, and this is giving the shape of spherical biofilm. There are then other influencing factors meant to be important for the formation of granules, and in this presentation I will show you how the presence of methanotrix, methanogenic cells, and the carbon source were really fundamental in our work with treating saline wastewater. So, as freshwater granules cannot be used to treat saline wastewater, we did start our granulation from the scratch, and our experimental work was done in different salinities. Here in this slide, I just show the highest salinity we did apply, 20 grams per liter of sodium. And we did start up our reactors with this anaerobic sludge from producing water treatment, which was actually already acclimated to 8 grams per liter of sodium. All our anaerobic tests were run at 35 degrees at increasing organic loading rates and fixed upflow velocity. And the reactors were fed with CP anaerobic medium, trace elements, and a substrate composed by glucose, acetate, and tryptone in a COD ratio 3:1. So we did observe the formation of granules already after 70 days, big format granules, and the process performance in terms of COD removal and methane production were really satisfactory. By applying several microscopy techniques, we were able to discover that these granules were rich in EPS, and that the rich EPS layer had as a major conjugate present a cloud-like EPS rich in mannose, which was always associated with the huge amount of methanotrix clusters that were present along the granules. And these methanotrix clusters were also serving as aggregation nuclei for the dominant bacteria, and in this case it's the Fluvitaleaceae filamentous bacterium, which was indeed aggregating around these clusters to favor granulation. Assessing the presence of a granulation key player like methanotrix, we did explore the effect of substrates changing. In particular, we did try to remove tryptone, which is known to be a stimulator of EPS production in many microorganisms. And we did run 2 reactors in the same conditions, but in one we didn't feed tryptone in the feed mixture. Reactor R4 here in the picture, and as you can see, while in presence of tryptone in reactor R3, we did form a granules bed after 40 days. In the case of reactor R4, no granules were formed, but instead a biofilm was growing on the reactor's walls, underlining that a complex proteinaceous substrate is essential to achieve granulation at high salinity conditions. Exploring the microbial community of the 2 reactors with and without tryptone by means of next-generation sequencing of 16S RNA gene, we did see that actually the granulation key players, methanotrix and the Fluvitaleaceae, were really negatively affected by the lack of tryptone, especially by the end of the process in reactor R4, where instead Enterococcus, a lactic acid bacterium known to be a HPS producer, and Thermovirga were taking over in the population. By also observing the biofilm biomass by fluorescent hybridization, we did see that methanotrix cells were well aggregated, but no bacteria were well aggregated as we were observing in the other granules. So apparently tryptone is fundamental for granulation at high salinity, but how much tryptone is needed to observe granulation? To answer this question, we did run 3 different upflow sludge blanket reactor. One feeding the same amount of tryptone that we fed in the other systems. In this case, is 16.77% of the total COD intake. And indeed, we did observe formed big granules. The same granules were formed where we were feeding just 1.8% of trypton, but biomass was accumulating in the settler. And in reactor 3, with just 0.45% of trypton, we did observe again biofilm growing on reactor walls and just flocks developing. Looking at the microbial community developed in the 3 reactors fed with different concentrations of trypton, we did observe that Enterococcus was the dominant bacterium in all the aggregated biomass, while Methanothrix in reactor 1 was likely outcompeted by acetate utilizing bacteria like Lentimicrobiaceae. Thermovirga and the Fluvittariaceae in reactor 1 had a good percentage in granules, while in reactor 2, the Fluvittariaceae were mostly washed out, even if their presence in granules was significant. In reactor 3, interestingly, Methanothrix had a relatively low abundance in the flocks, but it was the dominant microorganism in the biofilm. In conclusion, granulation at high salinity was possible and stable with a salt-acclimated biomass, but fed with a proteinaceous substrate. Methanothrix was the key methanogen in all the conditions analyzed, but the granulation was possible because bacterial groups which were always present in granules, as the Fluvittariaceae, Enterococcus, and Thermovirga, were aggregating on Methanothrix clusters. However, the same microbial community was not able to drive granulation without trypton. And we did establish indeed that a minimum of 1.8% of trypton out of the total substrate was needed to have granulation. And this was likely connected to the fact that resident microbes needs this substrate to excrete an EPS, which characteristics, most likely hydrophobicity, can favor granulation. And finally, I would like to thank all the people who were involved in this fantastic journey of this research line about saline wastewater. Thank you for your attention.
Thank you for the presentation. And is there any question in the online audience or here in the room? Yeah, David?
So what is the, again, the mechanistic role of trypton? Is it just a carbon source? Is it also a nitrogen source? So in the EPS, if you want to synthesize proteins, you don't need only carbon, but also nitrogen. So how can we track that then metabolically?
Thanks, David, for the question. Yeah, actually, this is something that we will further explore. We were running reactors in several conditions. For our understanding, and as we know from literature, trypton is known to be used also as a APS enhancer, also on pure cultures, for instance, of Enterococcus or bifidobacteria in industrial assets. So we were really thinking, okay, trypton is our granulation enhancer. Indeed, when we use, for instance, gelatin, we get granules, but way later in time. So we believe that is mostly what is needed is something that is a protein source, but should be partially hydrolyzed, but not also single amino acids, because when we were feeding just single amino acids, we didn't observe any granules. So we really believe that the status of this proteinaceous substrate, not only just a proteinaceous substrate, is really important to enhance granulation. Indeed, nitrogen indeed can be the reason why there is a granulation triggering, let's say.
Thank you.
But can I continue on this? Like in the cases that you don't have trypton, you still have EPS production, right? Because you said that you have flocks or biofilms, so it's kind of a specific type of EPS enhancer, because if not, you still produce EPS.
Our hypothesis indeed is that the difference is the hydrophobicity of this EPS, because we know that granulation is favored when the more the EPS is hydrophobic, the more we have the self-aggregation and granules formation. So our idea is that not only the microbial community is changing, but even if we have the same resident microbes, then the nature of these EPS is changing in a way that they prefer to self-aggregate or to aggregate to form a biofilm on the reactor's walls. So that is something that we still didn't explore, indeed. The nature of the EPS which is produced.
May I quickly ask here a question around, because tryptophan is also an important amino acid in the S-layer protein formation. And tryptophan itself is, of course, a very difficult molecule to produce biochemically, or difficult at least, costs quite a bit of energy. Could it be that it's not so much the hydrophobicity as a proper S-layer protein formation that is the limiting factor?
Well, actually, we did use tryptophan, which is a mixture of the casein hydrolyzed proteins from milk proteins. So this is a mixture of dipeptides, single amino acids. So we have really a mixture of things. So we cannot really say which ones of these compounds is triggering the granulation. But we know that this casein hydrolysate, for instance, contains also bioactive compounds, which are known to enhance the EPS production in these conditions.
Thank you. So we are now going to the last presentation of the session by Ramanujam Srinivasam from Singapore Center for Environmental Life Science Engineering. And we are going to launch the video now.
Hello everyone, I am Ramanujam Srinivasan Vedatri, presenting on behalf of my team from Singapore Center for Environmental Life Sciences Engineering, Nanyang Technological University, Singapore. Our research is on microbial protein production through mixed culture-based fermentation of soybean processing wastewaters. For a moment, can we think of this question based on the second Sustainable Development Goal, which is zero hunger? By 2050, the world population is expected to reach 9 billion, which will increase the demand for animal protein. Nearly 60% more crop production is needed to feed livestock and aquaculture. Such growing global food demand requires a fundamental reorientation in the existing food supply chain. So do we have a sustainable protein source to address the food security? Single-cell protein, SCP, derived from microbial biomass grown on nutrient-rich wastewaters, represents a promising protein source with high protein content along with vitamins, pigments, and antioxidants. Our recently published review highlights the single-cell protein production potential of microbial communities present already in wastewater. Food processing wastewaters are recommended as more reliable sources of single-cell protein due to their much lower content in pathogens, heavy metals, and other toxic contaminants. We also found more research is needed on the detailed chemical and microbial characterization of such wastewaters. The main objective of the current piece of work is to evaluate the microbial community-based protein production through fermentation of food processing wastewaters. Specifically, we would like to see how chemical and microbial characteristics of food processing wastewaters affect single-cell protein production. Identification of core microbial members involved in single-cell protein is also significant to understand the underlying principles. Finally, the whole approach becomes complete only when the produced microbial community-based SCP meet the animal feed requirements. Our study used 7 different batches of soybean processing wastewaters generated by soaking of beans in water. 4 bioreactors were started without inoculum, receiving different batches of soybean processing wastewaters. The subscript here represents the wastewater batch used for a particular reactor. 2 additional bioreactors were started with inoculum, receiving the biomass from one of the reactors that was run without inoculum. All of these are sequencing batch reactors of 4.5 liter working volume operated for 34 days. Both aerobic and anaerobic phases are included to favor the growth of aerobic and anaerobic bacteria. The dissolved oxygen was maintained between 0.2 to 0.5 milligram per liter, and the pH was measured and it ranged between 6 to 8.5. Let's first look into the chemical characteristics of different batches of soybean processing wastewaters. The panel here shows the variation of soluble total kaldal nitrogen (STKN) and carbon to nitrogen ratio (CN). CN is expressed as the ratio of soluble chemical oxygen demand to soluble TKN. The upper range of the soluble TKN represents the high quality of soybean processing wastewaters, and the lower range of the soluble TKN denotes the low quality of soybean processing wastewaters. To better highlight the variability in the chemical characteristics of soybean processing wastewaters, terms such as high and low were used in our study, considering the STKN levels. Then the question comes, how did this quality affect the single-cell protein production? The charts here show the variation of biomass protein content over the reactor operation period. Amino acid analysis using high-performance liquid chromatography is used to determine precisely the protein content inside the dry microbial biomass. Each of these panels look different from each other. At the same time, we see a variation shift individually as well. What is the reason behind it? It was due to the shift to a different wastewater batch. An increasing trend of biomass protein content was observed in some of these reactors due to the high quality of wastewaters used. A decreasing or a flatter trend of biomass protein content was observed due to the lower qualities of feed used. Hence, lower carbon to nitrogen ratio and higher soluble total kaldal nitrogen wastewaters yield a higher protein content. While there was a variability in the chemical characteristics of soybean processing wastewaters, similar microbial composition was found. Using 16S amplicon sequencing, Formicutes was identified as the most prevalent phylum. At a finer level, Lactococcus and Bicella were found as the most prevalent genera. Our next focus is on the microbial communities that got enriched in the single-cell protein bioreactors. They included Azospirillum, Gymobacter, Lactococcus, and Novus Figlobium. Two bioreactors that started with inoculum are represented here within the shaded rectangles. It's quite interesting to see how different genera got enriched in these bioreactors due to the different wastewater batches used. Microbial characterization is further complemented by fluorescent in situ hybridization technique, FISH. The pictures we see here denote the eubacteria represented in green and Azospirillum, which is our selected taxa, represented in orange. This matches very closely with our 16S amplicon sequencing data of Azospirillum obtained for the reactor on the chosen day. The final question is to check for the microbial protein produced for its animal feed relevance. The chart here represents the minimum essential amino acid requirement of aquaculture animals, trout and shrimp, represented in blue bars, and the green bars represent the microbial protein profile of the community-based SCP produced using several batches of soybean processing wastewaters. We can see either partially or fully the microbial protein fulfills the demand of aquaculture diet. The next step will be to use the produced SCP to feed the fish using aquaculture trials. The key message of the presentation is food processing wastewater can be converted directly into single-cell protein rich in amino acids, meeting the animal feed requirement through mixoculture-based fermentation. This approach does not involve sterilization of wastewaters. Hence, they incur lower production costs than the traditional single organism based approach. The study also identified relevant SCP producing taxa that can be plant growth promoting bacteria and certain probiotic potential strains that have been shown to produce vitamins B2, vitamin K2, and vitamin B12. I would like to acknowledge the work of the co-authors of this research, funding agencies, and many others who have contributed. Thank you very much for listening. I will be happy to take any questions.
Thank you for the presentation. So, is there any question online or on site? David?
Okay. Hi. Can you explain again what are the reactor conditions that you use to operate the protein production?
Sure. Yeah. Okay. So basically, our reactors are sequencing batch reactors. So they have both phases of aerobic and anaerobic. So the pH is actually measured and ranged between 6 to 8.5. So the thing is, like, we have maintained the temperature at 30 degrees Celsius and kept our dissolved oxygen level between 0.2 to 0.5. So these are all maintained at the same operation conditions. So every time the feeding takes place, we just look for the value of soluble chemical oxygen demand. Since the focus is not on treating the wastewater, our focus is on the microbial protein production. So we just wait for a certain level of soluble COD. So once it becomes lesser than the level, then we go for the subsequent feeding.
Then why Azospirillum was selected? So is it this alternation of aerobic and aerobic conditions or anaerobic aerobic conditions? So is it a denitrifier? What is the metabolism of this organism?
Okay, so Azospirillum is a nitrogen-fixing bacteria. So we see this abundance more coming up because of the quality of wastewaters. So the entire thing, the abundance, everything is linked to the quality of wastewaters we have used. So the wastewater is lower in nitrogen content. So this carbon to nitrogen ratio is quite higher. So we see the abundance of Azospirillum is because of the lower nitrogen content of the wastewater. So that's the reason we are seeing it for the Azospirillum. And the abundance we have shown it, so just to complement with our 16S, and it matches very closely with our data as well.
Shortly to conclude on my question, did you sparge the nitrogen in the reactor?
No.
During the anaerobic phase, how did you maintain the anaerobic phase? Did you sparge N2 in the reactor and maybe it enhanced the selection of this organism?
Thanks for the question. So this anaerobic is just a mixing involved. So we don't provide any nitrogen. So it is just a mixing phase. We just let the biomass to play around with the wastewater. Yeah.
Thank you.
Thank you, David. Thanks.
We can move maybe Mark to the panel discussion.
Okay, we can move to the general discussions, and also if there are specific questions for one of the presenters, you're also still welcome. Is there anyone online or in the room who wants to start the discussion? So if you're online, you want to start. Yes?
I have a question, Mark.
Yep.
Let me show my video here. That was potentially a bad idea. So I don't see anybody anymore. I'm sorry. I hope you can see me.
Yes.
Great. Okay. So I just have a comment, and it may be a little heretic, but it seems that a lot of reactor studies do not involve replication or repetition. And this is okay as long as we understand that the description of the microbial communities has to be observational. Yeah, so it's very difficult to link cause and effect if you don't have replication.
Mm-hmm.
Yeah, so this is of course one of the problems of running bioreactors. It's difficult to run several simultaneously. So that's I'm not sure whether anyone has here a comment on this, because it is good practice to at least see if you can replicate or duplicate your experiments.
That was my point.
if I can say something on this, Stefan,
Yes.
it's a very nice question mark indeed. I couldn't show all the reactors that we were running because we just had 8 minutes, but we basically replicated the same conditions in upflows judge bucket reactors many times. And every time, even if we were using the same biomass and the same conditions, we were observing a different evolution of the microbial community. Well, the dominant microorganisms, maybe they were almost the same, but I would question if we can say the word replicability when working on anaerobic reactors instead. I don't think that there's such a thing with natural mixed microbial communities. That's my impression. And again, we did run the same reactors with the same conditions for 3 years long with Danis Sudmaris, which was the PhD student, now assistant professor at Wageningen. I don't know if this adds something to the discussion.
So maybe I can.
That the dominant community was similar, but there was also a lot of, say, minor, which were small, if I understand well.
Yeah, in the sense that we were always developing a different microbial community in terms of the main protagonists and also the lower abundance groups. But we cannot say that each experiment that was run in the same condition was the same as the previous one. Never.
But that would mean a problem for you, because that means that whatever you conclude in the end has no value, because next time it can be different.
Who can say this? That's the question. Who can say this? Who can replicate?
So would you then say, if this is clear, true, that your conclusion was that if you add this tryptone mixture, that it will enhance granulation, that conclusion is only related to some erratic experiment, or was that reproducible?
It's enhancing always the dominant microorganism. We have always we have granulation, we have satisfactory performance in terms of methane production and organics removal. So this is what we can replicate, but we cannot really say that every time we replicate the previous process. That's what I mean.
Isn't it like we are storing the biomass in some condition, right, for a period of time? So there are two things here. One is the replicability, other one is the repetition. So if you are running the same reactor just at the same conditions, but we can't say it's the same biomass, right, because it's been stored for some times. So you're using it again. So maybe the question, if it is reframed as, if you are running the same reactors, maybe let's say instead of one reactor, right, you're running 3 reactors at the same time, then they're actually the replicates. So they should give us the same result because they're all operate at the same condition. So maybe we can take the discussion along that line. But I can just say something from my research, right? Because from the work I have shared, it's all so I don't run any replicates in my systems. They're all unique reactors because, as I was saying, each of them receive different batches of wastewaters. So, but I can share a bit of my work which I've been currently doing. So I'm running replicate reactors. As I was saying, they're all started at the same time. But the thing is, after some time, we are seeing some divergence in the communities. So even though they are the replicates, right, there might be the cases of bacterial succession coming in place, and we might end up in different result. To some extent, I agree partially with Christina, but what I'm saying is, still, out of 4 reactors, 3 of them are performing very similarly. One of them is behaving slightly different. So it can arise from the operational conditions, or it can be really due to the bacterial succession. So they need further investigation, but it's a good discussion. Thanks for Stefan initiating that.
David?
In this context, is there a way to decrease drastically the time of the experiment. So I remember when I ran reactors during my PhD, it took 300 days. We typically have profiles of 350 days and over 6 reactors, etc. Imagine the amount of work that it is, and I'm sure that everybody is doing that as well. Is there a way to do, maybe if we look for deterministic effects, maybe not the stochasticity that would need to reach a pseudo steady state, but for a deterministic effect to make an experiment over 2 weeks? And it means that we have to think about the design of experiments much ahead.
Okay. I can share again a bit of my insights, right? Because this is the first study we did with the food processing wastewater. We literally do not know whether we can produce biomass, which is our protein, directly from food processing wastewater. So later, we're able to get the inoculum and using it for our other studies. Then we were able to see significantly the reduction in the reactor operation period. One of the studies we operated around 100 days. And no one likes that, right? Because we can't stay with the reactors the whole day. So later, we're able to see it is able to reduce to a couple of weeks, 3 weeks, 4 weeks. We can get the same production rate because our focus is more on the production. So it is possible. And as you said, design is the key. So if we're able to crack the design, then we can reduce the number of days significantly.
Maybe to bring up some other aspect around experimental work in this field. Claudia Becerra mentioned it, and I saw Joe Wafer on Twitter supporting it, and that's the problem of metadata standardization and comparison. Does anyone have an idea? Because I think that's probably a point to really solve in this field. And does anyone would like to make some comments on it to see how we could standardize?
Hi, hello.
Yes, go ahead.
Can I talk? Okay. Yes, that was an idea because as we do this metadata analysis, we find that we have a lot of sequence deposited in the database, but and we have a lot of information, but we cannot use it because we have not standardized metadata, and probably the metadata is somewhere or in the computer of the author, but we cannot access. So I think it's important to do two things. One is to have a standardization of the metadata. So which are the metadata that we need to do some, that is important to run the reactor, as for example, the inoculum, the day of inoculation, the organic loading rate, I don't know, all this kind of thing, the composition of the wastewater, at least the higher composition as COG and so on. And the other is how to also to quantify other parameters, as the methanogenic yield, sometimes not the methanogenic yields of the methane, or the biogas, or concentration of organic acid, and so on and so on. I saw there was I saw in the I think it was in Delft, also in the last anaerobic digestion meeting, that was a special I don't remember. It was a group from France. I think it was Theodore Bouchet, that they were doing a database including metadata, and they want to standardize how to put the metadata. I can tell you our experience in the biohydrogen network, that we have to discuss a lot in this works that we make all together, how to put the metadata. So how to measure. So which things, for example, how do you express the yield of the process? By COD, by mole, or so it's important to have like a standardization of that, and then a repository of the metadata with the sequence, or with not the sequence, but if that we can share this metadata. I think it's important also to compare data from different works, because if we have such a variability in the process, in the different process, probably the variability is because also the microbial communities diversity, and also that it's not one microbial community that can do one process. There are several ones, and dependent of many conditions, you can select one or two. But if we have more data to compare, we can have more strong conclusions. And also to look at the function of the microorganisms, not only the names of the microorganisms, but the function, because maybe this difference in the microbial communities can be explained by the function of the function is the same. That's a lot of works doing in methanogenic reactors, and you run a reactor, or different reactors running in the same time, same way. They have different microbial communities, but the yield is the same. So I think we have to change our mind to look at functions and yields and compare that.
Thank you. Anyone else want to comment on this aspect, or shall we ask Martin in his lecture to give a few slides attention to this in the coming days? Okay, Sergio, you can handle the chat.
Maybe Joseph and Weaver, do you want to participate to say more or less what you wrote in the chat, so everyone can hear also here in the on site, that people are not really checking the chat?
Are you online, Joe?
Yes, I am online. I've just been yammering at my microphone. I had a hardware switch as well as a mute button. I'm happy to rant again like I was doing in chat. So metadata has always been an issue for bioinformaticians, and there's really no good way to solve standardization except by just continually improving it. You never get perfect. My feeling is, on a very immediate level, something that we can do is, as reviewers, editors, and authors, is just make sure that data availability does not become just more prevalent, but also metadata availability. That helps a long way, even if it's not standardized. If there is a even halfway well-written document explaining what this data is and how it was taken, it can help with at least manual meta-analyses. Claudia also mentioned something that's just an axe of mine that I like to grind too, is looking at function and not just species name. And there's a great article, it's The Song, Not the Singer by Doolittle, that I recommend anyone interested in microbial ecology reads.
Can you share the article in the chat? It's
absolutely.
Just more in the chat. No further. Are there other people want to comment on this, or maybe think it would be possible to come up with guidelines that are helpful? Because of course this point comes back regularly, and the problem for most full-scale installations is that even if you get the data, the quality is not always that reliable, and that's what's one of the other hindrances.
If I can add something, Mark, just I totally agree with Claudia. And thanks for this very valuable opinion. I think before metadata, indeed, we don't use all the same methods to measure things in our reactor. So that's also the main issue. Someone is using a method for measuring carbohydrates or proteins. Someone is using another. We don't use all the same COD kit. Someone cannot afford COD kits, so they are making the reagents themselves. So this is, I think, the first obstacle to make such data comparable, indeed. And also microbial communities, of course. We all have our original inocula, which have different features, characteristics. And even if we use always the same inocula, it's like we want to ask to an athlete to run the Olympic Games and perform always the same way. I don't think it's always possible. So, but indeed, I am totally in favor of sharing data with other groups that can compare what we have, because instead of publishing and publishing, we should start understanding what we are producing altogether and how this can help plant operators. That's all. Thanks.
Okay, thank you. I would propose that we have a break now to have chats or go bio breaks, coffee, beer, wherever time zone you're in. We will be back at 4:30 Dutch time, and you are free to calculate what it is in your own time, because it would take too long to go through all the time zones. And so I enjoy your coffee or what else you're getting at this time of the day. And thanks to all the speakers in the session.
Thank you.
Thank you, Mark. Thank you.